Welcome, wonderful Da Vinci Bio students, to the review for EKS4 Energy. Just to give you an overview of what we're going to do, I'm going to go through everything that we've covered in this unit in brief. Trophic systems, energy flow, photosynthesis, cellular respiration, and fermentation. And feel free to stop the video or just use the wait time I'm going to kind of give you to answer questions. Think ahead to what I'm going to say. Also, there's going to be a study guide that's posted with this video on Edmodo. You can use that to follow along and answer questions while you watch, or use it as a review after you watch this video, or before. Up to you. So, first thing we're going to talk about is just the flow of energy. As you know, all organisms require energy, and there's a few ways that they can do it. Autotrophs, of course, are the ones that produce their own energy, hence the name auto, alone, by themselves. Trophs, troph means energy, and those are like plants, trees, flowers, blah, 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 and also some bacteria and algae as well. Autotrophs don't need to consume anything else. They just take in light and other nutrients and are able to produce their own energy. Some don't use light either. They use chemicals. They're called chemotrophs. Just a side note. Heterotrophs have to consume autotrophs or other heterotrophs in order to get energy. Those are like us, this cute little rabbit here, every other animal you can think of, and actually even some plants, like we saw that Venus flytrapper during class once. Trophic levels, we covered a few during that activity where you positioned yourselves and to show the energy flow. Producers are autotrophs. They're the ones that produce their own energy. Every other thing after that has to consume a producer or another heterotroph, of course. Primary consumers consume producers, secondary consume primary consumers, and tertiary consumers consume secondary consumers. And decomposers, finally, they take down dead organic matter and turn it into nutrients, or something that goes back into the soil so producers can take it up, or it's used for other greater things. Examples of an energy flow would be a plant, which is a producer, an autotroph, which is eaten by ants. These are leafcutter ants, and finches, which eat ants. So the ants would be the primary consumers, finches would be the secondary, and then this hawk, which would consume the finch. They're the tertiary consumer. There's nothing higher than them that can consume them. In theory, technically there is, but we're going to say they're the highest. You can have more levels of consumers as well. And then, these are worms. Worms take dead, decomposed organic matter and decompose them further, and so that trees and other plants and stuff can take them up. It's really cool because almost every, pinch of soil you see probably has gone through an earthworm so many times. Now to the good stuff. Photosynthesis. That's what plants do in order to create energy. Quickly, here's a tree. So if you zoom in, you get a leaf, and if you zoom in further, there's the cell of the leaf. And if you see there's little green dots. Those are the chloroplast inside of the plant cell, and that's where our wonderful process of photosynthesis takes place. This is a chlorophyll. It has two membranes, the outer membrane and the inner membrane. Both of them are phospholipid bilayers. This is really interesting because you can think about how why would they want to do that? Why would it be this way? And Mr. Choi mentioned in his video that one theory is that a bacteria way back when consumed another bacteria that had these autotrophic capabilities of creating energy, and then so there's one bacteria within another, and then maybe another one consumed the other one to become the cell. We don't know. But that's just a theory and something cool to grapple with. Now you've got the outer membrane, inner membrane, and the little disc-like things are the thylakoids and a stack of discs. So if the disc was a pancake, the stack of pancakes are granum, Granum, and multiple stacks are grana. Pretty cool. And all the empty space around the granum and grana is the stroma of the chloroplast. So inner membrane, outer membrane, space outside is the stroma, the little disc is a thylakoid, a stack is a granum, and multiple stacks are grana with an A. Oops, forgot that title. Okay, so this is the complete balanced equation for photosynthesis. Photosynthesis needs 6 carbon dioxides, 6 waters, and light, of course, to produce glucose, C6H12O6, which is a sugar, and as a byproduct, it produces oxygen, which is breathed out of the air by the leaves. Okay, let's briefly go through how photosynthesis takes place. Now, when the light hits the plant cell, it goes to the chlorophyll, which are inside of the chlorophyll, which are inside of the plant cells. And okay, I'm sorry. I'm totally losing it. Okay, the chloroplast in plant cells, where it hits the thylakoid, which in the membrane it has chlorophyll, which are the receptors of the light energy. And what happens is the chlorophyll starts to vibrate a little, emitting electrons, and then the electrons are passed down around to produce ATP. Which is passed to the Calvin cycle. And in this process, they also take NADP plus and bind with a hydrogen from water in order to make NADPH, which is also passed on to the Calvin cycle. So, because the water molecule gives off the hydrogen, the oxygen is given off. This is during the light reaction. Next, in the Calvin cycle, so that's here. So when light energy goes in, electrons are emitted, which bind with hydrogen from the oxygen, and so it takes the hydrogen here and the NADP plus and makes that into NADPH. In this process, they are able to create energy, which creates ATP, which moves into the Calvin cycle. Because the hydrogen gets used up, there's oxygen left over, which is expended as molecular oxygen. Okay, and then for the Calvin cycle, as you can see, ATP and NADPH go into the Calvin cycle. It goes through several different reactions to produce glucose and using carbon dioxide. And then during this process, the ATP is used up, so it takes off a phosphorus. Oh, sorry, I keep doing that. It takes off inorganic phosphate, which makes ATP into ADP, and the NADPH uses its electron, making it NADP+. It's important to note here that although water goes into the light reaction and carbon dioxide goes into the Calvin cycle, oxygen comes out of the light reaction only. So there's no direct connection between carbon dioxide and oxygen here. Okay, moving on. So let's cover this real quick again. So light reactions. What goes in? Think about this to yourself for just a second. What goes into the light reaction? What are the reactants? Think about the equation. Which part of the equation goes in to the light reaction? So first of all, of course, we've got the water, which is one part of the balanced equation, H2O, it needs to give the hydrogen in order for NADPH to become NADP plus, to become NADPH. Also, light energy, of course, we really need that in order to excite the electrons and make this whole thing possible. NADP plus goes in order to become an electron carrier, NADPH, in conjunction with water and the energy. And ADP goes in order to make ATP. Oh, I'm totally giving away what comes out part. But, okay, what comes out? If you think about what goes in, like the water, what does it contribute to? NADP plus, what does it contribute to? ADP, what does it contribute to? Take a second to think about that. Oh, I should do this Dora Explorer style. Do you know what comes out of light reactions? Blink, blink, blink. Ready? Oxygen. So oxygen comes out as a result of water using up its hydrogen. NADPH becomes an electron carrier, so NADP plus takes on an electron to become NADPH, and of course it takes the hydrogen from the water. And ATP, and ATP, which is an energy carrier. So make sure you note that NADPH is an electron carrier, whereas an ATP is an energy carrier. So light-independent reactions, also called the Calvin cycle. What goes in? Do you know what goes into the light-independent reaction? Blink, blink. So we covered water in the light-dependent reaction. What is the other thing in the balanced equation that we haven't considered yet? Right, carbon dioxide. Whoa! Also, what goes in that we made from the light reaction that goes into the Calvin cycle, don't forget about NADPH, which was made in the light reaction and is carrying electrons into the Calvin cycle, and ATP, which was also made in the light reaction and is carrying energy into the Calvin cycle for this process to be possible. And what comes out? So what is this whole point of photosynthesis? What are we trying to accomplish? Blink, blink, wait, wait. Glucose. We need to make glucose in order so that the plant has sugar, which they can turn into energy later on. So you might be thinking, oh, why don't they just go straight from light to ATP? It's because they need to make something that they can store energy in. And ATP is only useful in using energy right away. So you don't really store ATP, you store it in sugar, and then you make ATP when you need it. Just a side note, if you're wondering. Also, they make NADP plus because the electron gets used up in the reaction and from NADPH, hence making it lose the hydrogen and the electron, making it NADP plus. So if you lose an electron, which is negative, you get a plus. Also, it uses energy, ATP, which makes ATP lose one inorganic phosphate, making ADP, which only has 2 phosphates in comparison to ATP's 3. So sorry. Okay, so let's review this process again. Light goes into the light reaction along with water and creates ATP and NADPH, which we want to go into the Calvin cycle. ATP is created by light energy bouncing electrons around and allowing that energy, taking ADP, giving it energy in order to bind with another inorganic phosphate to make ATP. Water goes in and gives off its hydrogens, and along with an electron and the hydrogen, NADP plus becomes NADPH. And because it uses up its hydrogen, oxygen remains and is breathed out of the plant. Next, in the Calvin cycle, ATP and NADPH go in. They, along with carbon dioxide, they combine to make glucose. So in this process, NADPH uses up its electron, so the Calvin cycle needs to spit out NADP+, and ATP uses its energy, So one of the phosphates is lost, making ADP. Let's think about what would happen if you didn't have light. So no light. Think about which side of the reaction, the light reactions or the Calvin cycle, would be immediately impacted. Think. Okay, so since light energy isn't immediately needed in the Calvin cycle, once you put a plant in a dark room, right, it won't stop making glucose right away because there's still some ATP and NADPH that was left over from before. However, after some time, the whole process will stop because glucose, it'll stop making glucose too. But if you think about it, what will stop right away is oxygen production because you can't make oxygen without light. Next, let's think about what would happen if you didn't have carbon dioxide. Okay, so no carbon dioxide. Notice which phase of photosynthesis carbon dioxide impacts. It's the Calvin cycle, right? So if it only impacts the Calvin cycle and it doesn't really, directly, the first thing it's going to stop making is glucose. However, for a while, the light reaction is going to be able to make oxygen because it will have NADP plus and ADP that's left over from the Calvin cycle that was running previously. Once this runs out, this whole process will stop. Oxygen won't be made, ATP and NADPH won't be made as well. Okay, moving on to cellular respiration. So cellular respiration is the process in which the sugars, either that the plants that make or that we heterotrophs consume, are turned into energy in the form of ATP. Look at these people running marathons. They're using ATP like crazy. So cellular respiration takes place in the mitochondria. Mitochondria, like you learned last semester, is an organelle in the cell that is specialized in making ATP. The mitochondria, like chloroplast, has an outer membrane, an inner membrane, and it also has a similar theory behind it that bacteria consumed other bacteria. And if you notice, there's like a little space in between the outer and inner membrane. This is called the intermembrane space. Intermembrane. Okay, that's horrible. Anyways, you don't need to know the cristae, but know that this space inside of the mitochondria that's not the membrane is called the matrix. Okay, in cellular respiration, glucose is made into ATP. So, I didn't include the complete equation. I'm so sorry. So, let's write it now. C6H12O6 is glucose. And then plus CO2 yields 6O2 plus 6H2O. Oops. No, that's not right. That's right. That is so wrong. Ignore. 6O2 yields 6CO2 plus 6H2O. So, the opposite of photosynthesis. If you learn one of the equations, you'll probably be able to remember the other. Now, note here that there are several phases of cellular respiration, and we'll go through those slowly, so don't freak out. We'll be okay. Okay, so first there's glycolysis. Glycolysis takes place outside of the cell, so in the mitochondria, in the cytoplasm of the cell. So that's called the cytosol because it's not attached to any organelle. It's the cytoplasm that's not attached to any organelle, the cytosol. Glycolysis, cytosol, okay? Glycolysis uses glucose, NAD plus, to create pyruvate. NAD plus is the precursor of NADH. I know it's confusing because photosynthesis uses NADPH, whereas cellular respiration uses NADH. P plants, P photosynthesis, however, it helps you to remember that they're different. So NAD plus goes into glycolysis to help glucose break into pyruvate. If you can remember that glucose has six carbons, whereas pyruvate has 3 carbons, maybe that'll help you remember that it makes one glucose can make 2 pyruvates. Similarly, between here, actually, there's a reaction where acetyl-CoA is made. I do not like this graphic, so I don't like this graphic, so we're gonna go to we're gonna go to a different one. I apologize. Okay, so NAD plus goes into glycolysis to make glucose and 2 ATP. So when you use NAD plus, it spits out NADH as the electron carrier, and it makes 2 pyruvate. Pyruvic acid is pyruvate, don't worry about it. 2 pyruvate come out of glucose because glucose has 6 carbons and pyruvate has 3 carbons. Acetyl-CoA also has 3 carbons, so 1 pyruvate makes 1 acetyl-CoA. 1 glucose makes 2 pyruvate, which makes 2 acetyl-CoA. So remember that ratio, it's pretty important. NAD plus, so where was I? Oh, in glycolysis, NAD plus is used to carry electrons, so it will take an electron to make NADH. So you get 2 NADH out of glycolysis, and during this process you also spit out 2 ATP. Now after glycolysis makes pyruvate, and pyruvate turns into acetyl-CoA, the process moves into the mitochondria. The citric acid cycle, which is the Calvin cycle for our purposes, takes place in the mitochondrial matrix, which is the inside space of the mitochondria. It's going to take acetyl-CoA to make NADH, so 6 NADHs and 2 FADH2s. The numbers are not super important, just remember the relative amounts to each other. So citric acid cycle makes way more electron carriers than glycolysis, which is really important because it leads to the next section. It also makes 2 ATP. Pretty important there too. Okay, and then everything that we make here, all the NADHs, all the electron carriers, move into the electron transport chain, which is super important because that's where all the ATP are made. We're going to go through the electron transport chain in a little bit more detail. Oops. Sorry, ignore that, please. Ignore all this. Okay. The electron transport chain is in which NADH come in and drop off their electrons to the complexes. So don't worry about what numbers and such, but what happens is NADH drops off an electron, and by doing so, it loses the connection between NAD and H, so then it loses a hydrogen too. So the hydrogens move up and outside of the mitochondrial membrane. I also forgot to mention that the electron transport chain takes place in the membrane of the mitochondria. Then the electrons are passed along the cytochrome complexes along the electron transport chain until it gets to the last complex where it binds with hydrogen and oxygen. This is finally where we use our oxygen to make water. note that here at every complex, an electron carrier, either NADH or FADH2, is giving off an electron so that more and more electrons are going through the process, and then more and more hydrogens are spit out into this space out here. Okay, and then finally, after all the electron carriers have given their electrons, the space up there where all the hydrogen ions are gets so dense with hydrogen that the hydrogen ions are all going to want to go to the area which has less hydrogen, and that's on the other side of the membrane. So it's going to go through this ATP synthase complex, this thing, in order to get there. It's going to go through everything, and then during their move outside into the this is the intermembrane space, intermembrane. From to the inside of the mitochondria, they are able to make ATP with that energy of the movement through the gradient. So ADP combines with phosphorus to make ATP here, and all the electrons in there make a bunch of all the electrons. Every time an electron is dropped off by an electron carrier, hydrogens go up into the gradient, into the intermembrane space, which creates a greater gradient, which makes hydrogens want to go to the other side, and that's what drives this whole process. Okay, let's do this again. So glycolysis. What goes into glycolysis? Think about it. So there's glucose, of course, the one that we have to break down and that plants make. We also have NAD+, which gets turned into NADH, and ADP, which gets turned into ATP. Now, I kind of give that away again. And what comes out is pyruvate, 2-pyruvate to be exact, per 1 glucose, NADH, and ATP. NADH is the electron carrier, ATP is the energy carrier. Now that pyruvate, the 2-pyruvate we made out of the 1 glucose, gets turned into 2-acetyl-CoA in the Krebs cycle. I have no idea. I didn't graphic that right. But anyways, the glycolysis takes place in the cytosol of the cell, which is the outside of the mitochondria, whereas the Krebs cycle takes place in the inside of the mitochondria in the matrix. What goes in is acetyl-CoA, so 2 of those per 1 glucose, NAD+, because we're making more electron carriers, and ADP, and then we're able to make NADH, FADH2, and ATP. 6 NADHs, 2 FADH2s, and 2 ATPs per glucose, which means 3 NADHs, 1 FADH2, and 1 ATP per acetyl-CoA. Just know that more NADH and FADH2s are made in the Krebs cycle than the glycolysis. Next, everything that we just made goes into electron transport chain. And oxygen. So there's NADH, FADH2. Oxygen is used as a receptor for electrons at the end, and by combining oxygen, molecular oxygen, and electrons and hydrogen, we make water, which is expended as a byproduct. NADH, it gives off those electrons and therefore becomes NAD+. It also gives off hydrogen, which goes into that inner membrane space. FADH2 also does the same thing. And remember, those hydrogens that go into that inner membrane space go through ATP synthase in order to generate ATP. Okay, so note here that a lot of ATP are made from electron transport chain, whereas only 2 per glucose are made in the glycolysis and the Calvin cycle. It's really important to note that distinction so that you know why oxygen is so important. Because without oxygen, we can't have electron transport chain. Yes, we can make energy with glycolysis and the glycolysis, but without oxygen, we can't make this volume of ATP. So let's think about this for a second. If we didn't have oxygen, what would happen? So that's where fermentation comes in. That's what we learned at the very end. Hopefully you caught it in that East lab. So what happens when oxygen isn't there? when there's no oxygen, it can't combine with it can't take the electrons that have gone through the electron transport chain, so the electrons just end up sitting in the complexes. The hydrogens also can't be taken, but they can kind of sit in intermembrane space. That's not a big deal. And then what happens is that the NADHs can't drop off their electrons anymore and make NAD+. And same thing with FADH2s. So what's the big deal with that? So if there's no oxygen, that means that the oxygen can't take the electrons to make water and then regenerate NAD+. And that's where anaerobic respiration or fermentation comes into play. So since we can't use electron transport chain to make ATP, we need to use glycolysis. And we need to settle with that 2 ATP that it makes, although it might be a lot less than 32 or 30, however more the electron transport chain can make. So in this case, we're running out of NAD+ in order to make the to make glycolysis work to make more ATP. So what happens there is that no NADH can be made, therefore the process just can't happen. No ATP is made, pyruvic acid stops being made, and then electron transport chain stops being made. But we have this ATP and this NADH that's already made and pyruvic acid that's just sitting there that can be used. So evolution is very clever. It uses these two things that it has in order to go back and make glucose and make ATP, and this is how it works. Ding! Oops! No! You're not allowed to see that yet. Sorry. I'm just going to actually delete that. Okay, and this is how it works, is that the pyruvic acid gets changed into lactic acid for humans and ethanol for yeast. And in doing this reaction of pyruvic acid to lactic acid, NADH becomes NAD+. And then we're able to do this reaction again as long as we have glucose. Even if we don't have oxygen, we're able to regenerate NAD+ in order for this reaction to take place. Again, so smart. Evolution. Except evolution's not a person, so never mind. So this kind of ends our review. Sorry that it was so long. To make things better, I am showing you a picture of a grasshopper. Actually, these are called locusts. What's really cool about locusts is that they can consume the equivalent of their body mass in one day. That might not seem like a lot to you because they're so small, but if you think about it, humans consume the equivalent of their body mass in about half a year. So this is actually pretty interesting to think. Is it that they're just using a lot of energy, or is it that they're super inefficient in using the food that they consume? Something to think about. Thank you for listening to this video and watching it, and I hope you use it as a study tool only, but spend more time rehearsing in your head and on paper, whatever works for you, how cellular respiration and photosynthesis work, how fermentation relates with everything. Go through the study guide and make sure that those questions make sense to you and you can find answers on your own. And good luck on everything. See you next week.